Okay, my name's James Horne, and I run the Nuclear Magnetic Resonance Spectroscopy facility at the CSL. And I'm going to give you a little bit of an overview of the facility and 3 examples that I hope will be reasonably relevant to you. It's a technique for identifying chemistry, for identifying molecules. And it uses very large magnetic fields. Oh, and by the way, I've got too many slides, but most of them are fluff, so I'm just going to crank through them, so it won't be too bad. So we have lots of warning signs. If you come in on level 3 at the CSL, you will be reminded to check if you have pacemakers and so forth before entering into that building, because we do have very large magnets. That is the magnet for the existing spectrometer, now no longer containing all of this stuff, because 2 weeks ago that was all ripped out and completely replaced by new electronics and completely new innards into that magnet, bringing us into the 21st century at last with this particular instrument. It's very exciting for all of us down at CSL. And this is how I describe what a nuclear magnetic resonance spectrometer does. A molecule is kind of a bit like a guitar. It's a lattice. It's got lots of connections, and they're all electronic. And if you put the right sort of energy into it, and you've got it in a magnetic field, it will resonate. All of those little atoms will resonate. And they tell us something about chemistry. They also tell us something about the context of where they are. So we get different frequencies for different types of nuclei and different kind of interactions that go on within the molecule. That's the level that we need to go to today. Pretty simple. So there you go. That's a molecule. That's a big molecule. It's a protein, 21 kilodalton. It's like a guitar. If I put the right energy in, it'll resonate and it will give me information back about its identity and its structure. We do have changes, as I just mentioned. The old instrument has had its guts ripped out and new electronics added. We're also, within 2 months now, I hope, 2 to 3 months, getting a new 600 megahertz system. That is a larger magnet. That means more sensitivity. That means better resolution. It means we can look at smaller amounts of the small molecules in chemistry that might interest us in a natural products sense. It means we can look at much larger systems if we have an interest in biopolymers like proteins, nucleic acids and so forth. It'll improve throughput in other areas as well. It'll be particularly useful for getting proper structure characterization of natural products from plant extracts and so forth. It will also help us with some of these profiling type experiments, metabolomics type experiments. One update. At the moment, the imaging capacity, and I'll talk about the microimaging, is now defunct at the CSL until such a time as we add a new imaging capacity to the new machine. Plans are in train for that, but I can't give you a timeline for that at the moment. So there you go. We put energy in and we get music back. Lots and lots of signals that tell us something about a molecule. That's several molecules actually vibrating, telling me something about them. What can we do with it? Big long list of different things we can do, from characterizations to monitoring reactions and so forth, looking at molecules binding to other molecules, imaging, metabolomics, diffusion studies. I'm only going to talk about 3 things today in the short time we have available. And the first thing I'm going to talk about is solution state NMR of small molecules with an example. It's an example of a natural product characterization, polygodiol species. This little fellow here. And if you prefer the kind of 3D view of it, what we wanted to do with this was not only identify what chemistry we had, what the topology of this molecule was, but actually what was the distribution of these methyl groups around the either side of the ring plane in this particular structure. We wanted to confirm what we had, but also to figure out 3-dimensionally what's going on, what's the stereochemistry. It's been suggested that this extract from the mountain pepper could have useful biological applications, hence the reason for looking at it. It was given to me by Amos from the Menzies, along with Jason and Chris down in chemistry. Chris is now moved on. As a purified solution in a 5mm NMR tube. And I went through and gathered a few NMR data sets. Don't worry what they're called, but basically the process is, well, what frequencies have we got in there? Can I find them all? What hydrogens and what carbons are those frequencies? And then I look at the hydrogens and I say, well, which hydrogens are attached to which other hydrogens through the carbon network? Which hydrogens have hydrogens that are kind of close together in space as well? So that I can kind of put the topology together and I can put the overall structure together. To give you an example, for that particular species, This is like a fingerprint, a frequency map, where each signal is a type of nucleus in that molecule that feels the magnetic field in a slightly different way and so resonates at a different frequency. So that's kind of a unique fingerprint for my molecule from which I can get chemical information. Secondly, I look at the same thing but looking at the carbon signals. Carbon-13 is an NMR observable nucleus, and it's about 1% of the total carbon in your molecule. We can see that, we just have to run it for a bit longer. In the new instrument, we'll be able to see that much quicker. Then we want to know which protons are attached to which carbons. That's what this experiment tells me. I have a carbon frequency down, I have a proton frequency down here and I have a carbon frequency here. So that's a proton here that's attached to a carbon there. And I can go through and figure out which ones are CH2s because they're blue, and which ones are CH3s and CHs because they're red. I'm not going to tell you how that is, but it's very useful information. Then I can tell you that the proton that resonates at this frequency here is close to a proton that resonates at this frequency here. They are 2 carbons apart. This proton's on one carbon, and this proton's on the next carbon. That's helping me to trace around the molecule. This rather groovy looking experiment is very similar, but it doesn't care whether things are joined in the network. It only cares whether the 2 hydrogens of interest are close together in space. And like little magnets, they influence each other. So every green blob tells me that a proton at this frequency is less than 5 angstroms away from a proton at that frequency. So I know what's attached to what, I know what's close to what, and I can start making some decisions about what I have. I can assign chemical shifts to all of the carbon atoms in that molecule. That's frequencies. Chemical shift means frequency. I can assign proton information as well. I have a unique list now. I know what every signal in those spectra corresponds to in the molecule. So I can now answer the question about, well, what's the stereochemistry? Do these things point up? Do they point down? By looking at what things are close to what on either side of the ring plane. Because you cannot detect an interaction through space that goes through the ring plane. It's effectively a barrier. And I can then find that these methyl groups are close in space to these protons in this side of the ring plane. They're all on one particular side of the molecule. And I now know the stereochemistry of that species. If that's a very useful biological molecule and I'm going on to do chemistry in the lab to develop that as a platform for a whole development program, I'm now making the right thing that will interact in the right spots on receptors when you add it to the system you're interested in. What else can we do with it? This will be pretty quick. We could do microimaging, and we'll do again in the future. I'm not going to describe that. I'm just going to show you some examples. This is work done with Matt Calverley and Penny Measham looking at development of cherries. We were actually particularly interested in looking at cherry stems, but the fruit look a lot nicer. So they're about 20 to 30 millimetres wide. That's the limit of the size that we can go to with a microimager in the NMR. And these are sagittal sections looking through the fruit from roughly halfway out to the periphery. And these are transverse sections going through the fruit. And we looked at this over the course of development through the orchards. And you can pick out quite a lot of microstructure of the vascular system in there. And the darkest spot in the region where there's not much signal because there's not much water in the pit, if you like, at the centre. Another example, broccoli florets. That was one that we threw out last time here just to show, hey, to remind people that we did have a microimaging setup that people might like to use. Again, you can see some good details down to the 10 to 100 micron sort of level pretty reliably. With a new system, we'll be able to do things a lot more interesting things like looking at diffusion of solutions through these structures and site-directed spectroscopy where we can actually get sort of fingerprint reading information from specific sites in structures. And we can also fit a live mouse. That's cranial sections through a mouse. These are not very good images, partly due to my skill at the time that we were trying to do this. But that's another possibility. We can fit the head of a mouse into these systems and keep them alive as we do things. So hopefully there will be applications in the future for plant science when we get that up and running. The final thing I want to show, in the time I don't have available to me, is the idea of using NMR in a somewhat untargeted sense where we just look at a snapshot of the metabolic physiological state of a system. Metabolomics. That's human blood plasma. Very simple experiment. You just extract it, the aqueous component in this case, and you reconstitute it in NMR solvent, and you take a reading. It's probably about 200-300 different metabolites in that system. It's human plasma, and we can look at different populations. of that. In this case, this was adult males pre and post infusion with an intralipid substance, looking to see whether we could detect changes in those profiles. So we had a statistically significant number of individuals. We looked at the two profiles, and in the same technology that Thomas showed with the unscrambler, we looked to see whether we could see differences in those two profiles, whether they would cluster. And we can come up with some simple latent variables which make a nice discrimination between the two populations, pre and post infusion. Once we do that, we can go further than that. And we can look at libraries of known metabolite NMR spectra for humans, relevant to the field of the spectrometer that we have, and we can go searching to see, well, do those latent variables, do those residuals line up with known metabolite species? And then we can go through and look at the spectra and see, well, okay, this particular doublet at this particular frequency, 3.81, shows up nicely in the human metabolome database as belonging to beta-hydroxybutyrate, and it could also be confirmed from other frequencies within the same molecule that also show changes. Because NMR actually has a lot of information for each molecule. It might have many different signals for one particular molecule, and all of those might be reporting on the differences between your populations. So it nicely showed, in this case, changes in that particular lactic acid metabolism. This was another example using cultured cells from knockout mouse models, looking at neurodegeneration, knocking out in and out alpha-synuclein from the genomes of these mice, and looking to see what in an untargeted sense what changes there were. And then going through, this was done in parallel with Richard with LCMS, and this is just showing you the sorts of information that you get out from NMR and from LCMS of the various metabolites that have been shown to have changed between those two discriminated populations. It's quite nice to do both the NMR and the LCMS because you don't get complete coverage in either technique, and some techniques are better at picking up certain metabolites than others. So you get two orthogonal techniques agreeing, and that strengthens your case much more. Done. Thank you very much.